Hello everyone, my name is Maggie Bennewitz and I am an assistant professor in chemical and biomedical engineering. It's my pleasure to welcome you to West Virginia University. Now my lab has two main research projects going on. The first one is the development of novel contrast agents for the early detection of breast cancer. So we are specifically focusing on magnetic resonance imaging, or MRI, which is an imaging technique that utilizes strong magnets and radio waves to get a picture of water within the body. But to be able to visualize breast cancer, typically a compound called a contrast agent is injected into the bloodstream, and that contrast agent will build up in the area of a tumor to light it up on the MRI scan. But there's some problems with the clinically used agents in that they're always on, so they're always going to give you a signal regardless of where they are within the body, and they're not targeted. So these agents are going to build up in both benign, harmless tumors as well as cancerous ones. So what we really want to do is to develop a new kind of MRI contrast agent based off of nanoparticle technology that'll be activatable. So our agent is going to specifically turn on within breast cancer but remain off within benign tumors. So within this project, you'll be able to get a large range of skills. How do you design, fabricate, and characterize the nanoparticles? You'll be doing cell labeling studies. You'll be able to be exposed to microfluidic chip platforms where we can mimic the tumor microenvironment in vitro, as well as be exposed to animal models of breast cancer, in which we can evaluate MRI signal, the distribution of where the contrast agent goes, as well as evaluating any toxic effects. The second project that we have in the lab is involving e-cigarette toxicology. So we're using a different type of imaging called fluorescence intravital imaging, in which we can apply to be able to look at different kinds of blood cells and how they're moving in the blood vessels of the lung after the mice have been exposed with e-cigarettes. So we're specifically looking at clumping of these cells and what kind of downstream pathology that can cause within the lung blood vessels. I'm not actively recruiting for that project, but feel free to ask me more questions about it if you're interested. In general, with the lab, we do have weekly lab meetings where the students have the opportunity to present their research findings, as well as get feedback from me, as well as other lab members, ask any questions, also to be able to help brainstorming with troubleshooting different kinds of project ideas. And I really like it when students are creative and they take the reins of their project and try to figure out where they think the project should go. So if you would join my lab, I would encourage you to do the same. As well, when you're a part of my lab, you'll be able to attend national and international conferences to present your work, as well as be actively involved in the preparation of peer-reviewed manuscripts so you can get your research out in published form as well. So if you are interested in learning more about my lab, I definitely welcome you to approach me to be able to talk more about our research and what's going on, and I hope to hear from you soon.